Yeah. Thank you for a great introduction. My name is Lee Joon, as Greg mentioned. I also go by LJ. I have no preference for either. And it's been about a year since I joined the SIBYLS beamline as a postdoc. I've been primarily working with lipid nanoparticles, really, and using the high throughput SAXS platform to collect a lot of different data sets. I'm not gonna go into too much of like the lipid nanoparticle analysis in this talk, but rather I'm introducing a tool that I've been starting to use like more and more because of the sheer number of data sets I have to work with. And I'm hoping that this will be as useful for you as it has been useful for me. So let's get right into it. I'm gonna pull up my laser pointer. Okay. So we've heard a little bit about the high throughput SAXS platform from Greg earlier in the morning today, but we can generate data very quickly at the SIBYLS beamline, particularly if you use this capability. And so a full 96 well plates, it's about an hour and 20 minutes, I want to say. And so really the bottleneck at that point becomes data analysis. So maybe you ran a screen, like drug screening that Greg mentioned earlier, or maybe you're varying something in the buffer, you're adding like excipients into your system, and you want to look at like how all of your data sets that you generate are related together. And I personally would not want to plot all of them on raw and look at them individually because that would be a lot of time and maybe eye strain. And so this is a nice tool for that could help you illustrate how the SAXS data sets are related to each other. So it's called SAXS Similarity. Greg published this paper back in 2013 with the help of Kevin Dyer, and it's on Nature Methods. And it's a web application that gives you an idea of how the SAXS data sets are related to one another. And this is the interface that you will see. And I'm going to be doing a live demo of this. We will see this again in a little bit. Couple highlighting the main features. It generates a SAXS structural comparison map that quantifies a pairwise agreement between two data sets. And it generates this type of a matrix that's also similar to like a heat map. And how you read this is the blacks in the middle are self-comparisons. So comparing data set number 1 to data set number 1, data set number 2 to 2, and so on. These two either sides, and I realize it's really hard to see my laser pointer because these are all red, they should be symmetrical. So I typically deal with like this top half of the square. And so for example, this cell right underneath my laser pointer will be comparing data set 1 to 2, and that's like bright red. And on this legend on the right, it shows up as like high similarity score. Whereas compared to this cell, maybe comparing data set number 1 with 8, this is more like a yellowy, so that's like low to medium similarity. And you can actually see this better. If you were to click on these cells, it will show you the two scatter plots that it's comparing. And so this yellow one that's not so similar is the top plot. And these are the two scattering profiles, and you can visually tell that these aren't very similar to each other. And then if you were to click on some of the bright red cells, then it would look something like this on the bottom, where there's a nice overlay of the two scatter plots. The metric that we are using for the web app is volatility ratio, which some of you, if you like finances and stock trends, are probably familiar with. And we have developed a volatility ratio metric for SAXS. And it's been really nice because it helps us pick up the distinctions and trends in the data sets more easily compared to some of the other more well-known values like chi-squared. Like a big difference I would point out, for example, in chi-squared is this deals with sort of the subtracted difference in the SAXS data sets, whereas the volatility ratio deals more with the ratio. So then what happens is, Greg mentioned earlier, in the low Q region, there's like high intensity of scattering. So if there's like a difference there, that tends to dominate all of the results. An example of these different metrics that we can use is illustrated in the figure right here. When we compare the reference data set, which is like all closed conformation of the ATPase with another all closed ATPase, so this should be like identical, basically, like super high similarity. And as we slowly increase, as we like slowly move towards like more of an open conformation for the ATPase, we would expect the similarity index score to become smaller. And we see that really well in the VR. This is a third row, because that gradient of color, like decreasing, going from high similarity to low similarity, is really like nice and smooth. But you see that's not as good in the case of like chi-square and like Pearson coefficient. And this is consistent in the right side, that's looking at the classic state ribosome versus the ones with like swivels and rotations. And you can also see a similar trend here. So that's why, like, for the web app that we have up, the volatility ratio is set as default, but you still do have the option of changing between these methods if you choose to do so. The other main feature I will highlight is the forest plot, and that also helps us visualize similarity in a different manner. So here, each of these circular nodes is a different SAXS data set, and that size of the circle is proportional to the radius of gyration, which now you have heard about many times. And so this one is a little smaller compared to this orange one on the right. So the radius of gyration for the blue is smaller. So this would be like a protein that's like in like a more of the closed conformation versus the open. And the distance between these circular nodes are proportional to the degree of similarity between the data sets. So if they're closer together, like these ones sort of like right next to each other, these are more similar compared to the ones that are really far away, like these two on the other sides. So this helps us get a nice view of sort of the trajectory of your sample, like how it moves and how it changes under like your changing conditions or whatever you're screening. So those are sort of like the really big chunks of the features that I will highlight from SAXS Similarity, but I also wanted to talk to you a little bit about some applications of like why you might want to use this for your purposes. Since the original 2013 paper, SAXS Similarity, that Nature Methods paper, has been cited quite a bit, and for a lot of different research areas. So these are just a couple papers that are pretty recent that I wanted to highlight. And it ranges from like protein design to DNA repair, self-assembly of protein cages. Just to talk about like my personal interest when it comes to using SAXS Similarity, I use it mainly for like clustering large groups of data, which saves me a ton of time. So again, I'm not going into too much detail, but essentially what happens is my collaborators will give me like 3 or 4 different 96 well plates all at the same time, which then gives me like a couple hundreds of data sets to work with. And so using SAXS Similarity for me has been a really nice visualization tool to figure out like what kind of strategy I'm working with in going about analyzing all of my data sets. So for example, this one is a very small portion of like one of my plates. And you can already see that there are different clusters of that you can observe, and I've highlighted those in these yellow blocks. And they match up with the different conditions that I was trying to look at. And so using this, I'm able to come up with some sort of a strategy, like maybe for conditions 1 through 3, I'll use one type of data analysis platform. And for condition number 4, I'll use something different because these data sets look and behave sufficiently different compared to all of the other ones. The other, another application that I found, this is not mine, but I thought it was really cool, was validating protein engineering designs. This was back in 2015 by the Baker Lab, and they came up with computationally designed like 80 different proteins with different stackings and torsions, and et cetera. And so this is like a small subset of those designs. And they were wondering how many of these topologies that were currently not observed in nature could be actually like stabilized and collected data from experimentally. So they use the high throughput SAXS platform and collected lots of different data and analyzed it using SAXS Similarity. So going over how to read this big heat map, this top left portion is comparing experimental data to experimental data, and the top right and the bottom left are comparing experimental data to calculated data. And then the bottom right would be comparing calculated data to calculated data. The part that we would be most interested in, because we're trying to match up calculation versus what we actually saw in experimental results, is this calculated versus the experimental portion. And so that's like sort of blown up on this figure right here. And of this, the region that we're particularly interested in is this diagonal line that I've drawn in black. And this is essentially comparing the calculated data from sample 1 to experimental data from sample 1. So really being able to look at all of the different protein designs and seeing how well the calculations match up to the experimental values. And so if there's high similarity on this diagonal line, that means the experiment matched our calculations. And if they are not red, like if the similarity is low, that means the experiment didn't really match the calculation. So again, really nice way to visualize a large number of data sets. And if you did see something that sort of threw you off, like you weren't expecting the experiment and the calculations to not match up, then you can pinpoint where you want to have like a more in-depth look and do further investigations. Okay, so that was pretty much it for all of the actual applications and feature highlights. So I'm gonna talk a little bit about, oh, it's like lagging a little bit. There we go. The tutorial data sets that I'll be using to go through the live demo. The first one is morph.zip, and this is of a protein that's undergoing the conformational change from open to closed, separated by 21 different steps. And these files are randomized where it's like frame 00 is like step 2, frame 01 is step 16. And we have this exact same protein, but under different conditions like salt concentration and pH under experimental data. So these two I will show in the live demo. This third one is the ribosome data, and that compares 24 large subunit entries on PDB. And I'm not actually going to go through this, but if you go visit the web app, you can download this on your own and sort of play around with it on your free time. So I am going to start the live demo section. I have the link to the SIBYLS website, but you can always Google it and it will take you to our website. And I think I have to reshare my screen because, there we go. Okay. Let me also know if I'm going too fast, or if you have any questions, just interrupt me, it's fine. So I'm gonna hover over the web apps. There's a lot of different options here. I'm gonna click on SAXS Similarity, and this should look familiar because you saw this already in my slide. I'm gonna click on this file, or this gray area, to open up a file upload. But you can also open a directory and just like drag and drop your files in there to upload all of your files. So this is the morph.zip. So undergoing conformational change. And it's gonna upload all of these frames and generate that heat map that we saw initially. So this is what it looks like when it first uploads all the files. Right now it's in the order of how I uploaded these files. But if you're not happy with how these files are ordered, you can always manually change them if you like drag and drop them, and the heat map will sort of adjust to reflect that change. Other really small features that I found I use sometimes, like the color range you saw earlier. I was using like a blue, white, and red instead. So this is like that option. The default is the yellow to red, and we also have the grayscale. This is the part where you can change the method, like your metric from volatility ratio to something like chi-square. I do use this sometimes just to see how it compares with the volatility ratio. One useful feature that I will highlight again is if you click on one of these cells, then it brings up, you can scroll down and actually look at the two scatter plots it was comparing, and I've shown you a screenshot of this earlier. Let's say that your data set is experiencing some sort of like a radiation damage, so you have to like do an earlier cutoff, then you can just use the slide bar to adjust for that range, and then that will get reflected on the heat map. Sometimes, especially for my own data sets, a lot of times the features are kind of centered around a very specific region. So then a lot of times my ranges tend to be a little more narrow. But for this demonstration, I'm going to keep it to default, which is Q of 0.015 to 0.2. And the top right of this little bottom section also gives you the radius of gyration for each of the frames, and also the difference between the two. And that number will also show up on the cell itself when you click on them. If you would like the numbers for all of the cells to show up, you can go to operations and click on show RGs, and these will be all of the changes in like the RGs between the two SAXS scattering profiles it's comparing. If you don't like that, just click it again and it'll hide it. One of my favorite features is the cluster option. That's what I typically use before I look at any of my heat maps. And that's gonna really arrange all of your frames or your data sets into order, going from like high similarity to like low similarity. So for example, in this top row, frame 19 is most similar to data set number 2, which is frame 20. And it's like least similar to data set number 21, which corresponds to frame 00. The other thing that's useful is this metric matrix. If you click on this, it might be a little hard to see, but I'm gonna just do that for visualizing. And this is showing you the volatility of ratio values for each of the cells. So if you are not using the heat map itself, but you still want the volatility of ratio values, then this would be a nice window to pull up. And the other feature that I've highlighted in my slideshow was the force plot. And this probably looks really familiar because I used the exact same figure to make my slide. So this again is undergoing the conformational change from open conformation all the way out to the closed conformation. So this is a really nice way for us to like sort of track our sample as it changes in our system. So if I were to sort of clear out this file and then load up the salts and pH experimental data, that would look a little different. It's gonna generate a nice heat map again. Again, it's not clustered right now, so I'm just gonna go ahead and do that just to get a nice idea of like what data sets are actually related to one another. And already sort of similar to my example of how I used the SAXS Similarity earlier, there are like two main groups that's pretty easy to tell for us. There's this top left portion that's like in the reddish region, and then this bottom right that's like sort of a smaller square of the high similarity data sets. And from the heat map, if we look at what kind of data sets these are all like, so this number 100, 300, 500 is like salt concentration. And the following number like 10 and 11 is pH. So these are all like high pH values. So we can already sort of see from this figure that the protein under high pH behaves very differently compared to the rest of the conditions. But even within this really big chunk of red region, it's kind of hard for us to really tell what's going on. And so I'm going to click on the forest plot again to give us a better idea of how sort of the little clusters might exist and how they might be related to one another. And so this is what that would look like. This bottom like chunk here are the high pH values you can see on the legend in the left side. These are all pH 10 and 11. These ones are all of the rest of the data sets, and we do see that these smaller groups of data sets are related to one another. So this is something that was a lot more difficult for us to tell using the heat map, but definitely way more visible using the forest plot. In this website, in the web app itself, it also gives you sort of a tip saying that if you were to combine the morph and the experimental together, it could give you a deeper insight into what the protein is doing. So I'm gonna actually load all of the data sets that I've just showed you together in like one go. That will be like 40 something data sets that are being compared at the same time. And so the heat map that gets generated is going to be pretty big. The cells are now really small. I'm going to, once again, click on the cluster and have it sorted out. And again, it looks pretty nice in terms of gradient, but it's even harder to tell what's going on because of how small the cells are now. So I'm just going to go straight to the forest plot. So now we see that this combines what we saw earlier with the conformational change going from open to close, but also with the experimental data under different salt concentrations and pH. These ones, the RG is actually a lot bigger than the ones in the open conformation. So these are sort of the aggregates that occur with high pH for these proteins. And these smaller ones correspond better with the actual like conformational change that occurs as the salt and the pH changes. So I would say in terms of live demo, that's like pretty much all I had in mind. If you scroll a little bit to the bottom of this, there are links to the original paper. Please cite this original paper if you try this out and you find that it's useful for your research applications. And then these are where some of the updates might be found if there are any updates. I think my recording will be available, but if you want to listen to Greg talk about this tutorial, then there's the YouTube video you can watch. Couple other smaller details like instructions, like file formats, data formats. And all the way at the bottom here are the 3 example data sets that I introduced you to today. So we went over sort of example 1 and example 2. Even though you saw me doing this, it would be still like a nice practice for you to download and try this out on your own. And definitely try out the ribosome data set in example 3 and see what you can do with this. And so that I'm going to stop sharing this screen and sort of go back to my presentation screen because I have a last slide in there just to thank. And yeah, so thank you all for your attention, and I'll take any questions if I can answer them.